Today we will talk about glycogenesis. Let's define it together. Glycogenesis is the storing up of sugar present inside the bloodstream into the liver in the form of a glycogen. First of all, in order for glucose to be stored inside the liver, it should be internalized by the liver, which means enters inside the liver. For that reason, we need GLUT2 receptors, a receptor that are present on the surface of hepatocytes that will facilitate the entrance of glucose from the blood into the liver. Once the glucose enters the liver, an enzyme named glucokinase will convert the glucose into glucose 6-phosphate. So from the name kinase, which means addition of phosphate group, this enzyme will add a phosphate group on carbon number 6 of a glucose molecule, forming glucose 6-phosphate. In contrast to glycolysis, this step was catalyzed by hexokinase. So what's the difference between glycogenesis and glycolysis, knowing that the same step is being catalyzed? Alright, keep in mind that whenever we are talking about storing up sugar, a glucokinase will be involved. However, when we are talking about on-spot usage of a glucose inside the glycolysis for ATP production or whatever reason, hexokinase will be the activated enzyme. Why? Maybe you will ask yourself, why that change or that difference in enzymes, knowing that we are converting the same product, glucose, into glucose 6-phosphate. Okay, let's imagine that you ate that specific sugary food and the glucose concentration increased in the blood, where the glucokinase directly released inside the bloodstream and stored all the glucose consumed. What will happen? Simply, there will be not enough glucose in the blood, not enough glucose to be catalyzed by hexokinase inside the glycolysis for ATP production, which means hypoglycemia and an array of problems will arise. For that reason, the body regulated the release of glucokinase after a couple of hours following the consumption of glucose, for the aim of giving hexokinase the chance to catalyze some of the glucose, activate the glycolysis, and produce the ATP needed by the body. After that, the excess sugar is still present in the blood. Glucokinase will store them inside the liver, hence reaching the equilibrium needed inside the bloodstream on the basis of glucose concentration. Hope I make it clear for you and you understand why we used glucokinase instead of hexokinase inside the glycogenesis. Great. Now glucose 6-phosphate is obtained, where another enzyme named phosphoglucomutase will mutate the position of phosphate group from carbon number 6 into carbon number 1, forming a compound named glucose 1-phosphate. This conversion is really important because glucose 1-phosphate structure will help in the synthesis of the upcoming products. Amazing. Now, when glucose 1-phosphate is obtained, this compound will help in the synthesis of pyridine diphosphate glucose, one of the most important compounds in this pathway, known as universal donor of glucose. This step is catalyzed by the enzyme UDP glucose pyrophosphatase, so from the name phosphatase, which means removal of phosphate group, where here kinase addition, here phosphatase removal of phosphate group, where this enzyme will remove phosphate group from glucose 1-phosphate, yielding the core, the glucose molecule. But because this dephosphorylation needs a source of energy, this phosphatase will remove phosphate group from a UTP molecule, yielding iodine diphosphate that's present here, and an inorganic pyrophosphate group. This inorganic pyrophosphate group will enter the cytoplasm, be converted into inorganic phosphate group, hydrolyzed by water, and produce the energy needed by phosphatase for the dephosphorylation of glucose 1-phosphate molecule, where here the remaining glucose, the core, will be merged with the iodine diphosphate obtained from the hydrolysis of UTP molecule, forming the iodine diphosphate glucose. Once iodine diphosphate glucose is obtained, another enzyme named the glycogen synthase will start to synthesize the glycogen core from UDP glucose molecule by extracting that glucose part. And once the core is synthesized, sugar residues will start to be added in a linear form for up to 8 sugar residues. Once we reached 8, a branching enzyme will be activated and cut from terminal side up to 3 residues and branch them as shown in the figure below. Keep in mind that sugar residues are linked by alpha 1,4 glycosidic bond Exceptionally, the first sugar residue of the branch is linked by alpha 1,6, which means carbon number 1 with carbon number 6. And by this, we form the extra complex form of a glycogen known as a glycogenin, ending the pathway of a glycogenesis. Hope I make it clear for you. Don't forget to subscribe, and I will leave links in the description for other YouTube videos. Thanks a lot.